Good afternoon, everybody. So today we are going to see a topic on molecular biology that is called as transcription. So basically what transcription means, it's given here. So it is a molecular process in which an RNA molecule is synthesized from a DNA template, and both the strands are identical, and of which one strand which synthesizes the RNA is called as the coding strand. And if you see, the nucleotides in this coding strand are complementary to what is called the template strand, and RNA is manufactured within cells by a process known as transcription and with an enzyme called as RNA polymerase. The transcription begins from the 5' end and it finishes at the 3' end, and what is made is called a transcript. So we'll go with the next slide, in which we have the machinery of the RNA polymerase. So we have the enzyme, a very powerful enzyme called an RNA polymerase, which will maintain or make an RNA. So for prokaryotes there is only one single large enzyme, whereas for eukaryotes there are three different specialized polymerases. RNA polymerase 1 makes ribosomal RNA, polymerase 2 makes mRNA and small nuclear RNA, and RNA polymerase 3 manufactures transfer and 5S ribosomal RNA with respect to eukaryotes, while in bacteria it is going to be a single enzyme. So there are small differences between replication and transcription. In transcription, a new form of nucleic acid called as an mRNA is formed, and it uses ribonucleotides instead of deoxyribonucleotides. While DNA replication needs a template, RNA replication does not need that, and the enzyme can begin transcription at sites known as promoters. And as and when the enzyme is going to add a ribonucleotide, the length of the chain increases, and one full entire mRNA is produced, which will be translated to a protein itself. And you must remember that with respect to bacteria, there can be many transcripts from one single gene. We go to the next about the RNA polymerase. So the enzyme has been purified from bacteria, and it is a multimeric enzyme. It has many subunits, of which the two alpha subunits will catalyze the synthesis process. We have a beta and beta prime subunit. So the enzyme having all the subunits is called a holozyme, but the holozyme is actually inactive, and to begin its activity it needs an initiation factor that is called as Rho. So hence the complete enzyme is actually having all the subunits plus the initiation factor. So we go with how transcription will begin. So usually transcription begins at one region called the promoter, which we see here. It has the -10 and the -35 regions, and the whole RNA polymerase kind sits on the promoter and begins laying out its first nucleotide at +1 position. We are going to see about the transcription as such. So RNA polymerase can unwind and rewind the RNA, and it holds the separated strands. It adds ribonucleotides to the RNA chain, and it is quite a fast enzyme. So the biochemistry of this, so the enzyme adds an RNA molecule or a ribonucleotide to an existing primer, and once a ribonucleotide is added to it, the length of the RNA chain increases, pyrophosphate is released, and the release of the pyrophosphate actually powers the enzyme, and it can do continuous rounds of transcription. So three steps of transcription are there. One is the template recognition, initiation, elongation, and termination. Well, the first step is not actually a part of the transcription. It is to identify the promoters. The next steps 2, 3, and 4 are going to be more critical. So we go with the first one, how the promoters are going to be recognized. So the DNA should be unwound at one point, and they should be making free so that the RNA can find them. The polymerase can actually be seated at a place which is known as the promoter region. So the promoter is actually a sequence of the DNA in which the RNA polymerase binds, and it can start the transcription as such. So from the polymerase start site, you call it as a promoter, and there can be many promoters for one particular gene. So as we see here, this is how promoter has two elements. One is the -10 box, which we know is the comprised of the nucleic acids TAT, and it is seen in most bacterial promoters. It comes 10 nucleotides upstream from the transcription start site, and we have -35 sequence, which is -25 nucleotides upstream from the transcription start sequence, and it has the sequence consensus called as TTGACA. Both these sites are required for binding of the RNA polymerase. So in the initiation step, what happens? The DNA is opened up and the promoter is identified, and the open promoter complex is formed, and usually at this point what is called a transcription bubble happens. So the DNA is transiently opened and one of the strands is going to be used for the transcription process. In this picture we can see that the mRNA has been already formed and the 5' end is actually getting free, and the enzyme is actually catalyzing the transcription. So here we have seen a picture again. So the sigma factor is usually required for the RNA polymerase to begin its transcription. So without the sigma factor, transcription does not start, and once the sigma factor is bound to the RNA holoenzyme, transcription will begin, and then it can be used again and again. And this is what is called as a transcription cycle. So at the start of transcription, the promoter is identified, the sigma factor binds along with the core enzyme, transcription is started, it continues till it reaches the stop site that we can see here, and once transcription is started, the sigma factor disassociates. It is again recruited during another phase of elongation, or what we say as transcription itself. So chain elongation, what happens? New ribonucleotides are added to the growing chain, and even at this point, that RNA template is actually still newly formed RNA is still bound to its DNA template, and the RNA polymerase seals all the nucleotides that are downstream from the transcription, whereas it unwinds all the nucleotides ahead of the transcription and upstream of the transcription start point. So we can see here ribonucleotides are added, and one activity is that the proofreading activity is still there to some extent even in RNA polymerase. This is the last step in which RNA polymerase is going to complete its job. It is called as termination, for which we need two kinds of terminators. In bacteria we have the row dependent and row independent terminators. In row dependent terminators, a row protein is involved, whereas in row independent terminators, actually the row protein is not much involved. And we have two kinds of things like this. So in row independent, a stem and loop structure is formed. The RNA polymerase will follow from it, and in the row protein, in this particular picture, we can see row dependent transcription, in which the row protein will recognize the last part of the mRNA, and it will form a complex like this, cutting off the RNA template from the DNA template. And these are two modes of transcription termination, namely row dependent and row independent termination. And